Hello. Today we will talk about quality control of creams and ointments. What is QC? The GMP component known as quality control is concerned with sampling requirements, testing, organization, documentation, and release procedures that guarantee the necessary and pertinent tests are carried out and that materials and products are not released for use or supply until their quality has been determined to be satisfactory. What are the steps of quality control in the pharmaceutical industry? 1. Raw materials testing. The first step in quality control is to test the raw materials that will be used to make the drug. 2. In process testing, once the raw materials have been approved, they are sent to the production floor where they are transformed into the finished product. In-process testing is conducted at various stages of production to ensure that the drug meets all quality specifications. 3. Final product testing. The last step in quality control is to test the finished product to confirm that it meets all the required standards. Only products that pass this final testing stage are released for sale. Let us talk about semisolids. Semisolid type. A. Creams. B. Ointment. Differences between cream and ointment. Ointment is oil-based, and cream is water-based. Ointment absorption is slower than cream. Ointment stays longer time on the skin than cream. Ointment is thicker consistency than cream. Ointment is greasier than cream. Advantages and disadvantages of cream and ointment. Advantages are: 1. Self-administration is possible. 2. By removing the transdermal patch, the drug input can be stopped at any time. 3. Permits the efficient use of medications having a brief biological half-life. 4. Permit medication administration with a limited therapeutic window. 5. Provides regulated plasma levels of highly powerful medications. 6. Drug input can be stopped right away if toxicity arises. But their disadvantages are: 1. Skin irritation. 2. Skin sensitization. 3. Uncomfortable to wear. 4. Relatively poor patient compliance. Let's talk about properties of semisolid dosage form. First, physical properties. Smooth texture, elegant in appearance, non-greasy and non-staining, and non-hygroscopic. Second, physiological properties. Non-irritating, do not alter membrane, miscible with secretion and low sensitization index. Third, application properties. Easily applicable, and high aqueous washing ability. Fourth, storage properties. Not exceed 25 Celsius degrees, stored in a well-closed container, and not be allowed to freeze. Now, let us talk about QC tests for cream and ointment. Here, we have the classification tests for cream and ointment. 1. Universal tests. 2. Specific tests. 3. Performance tests. 1. Universal tests. A. Description. This includes visual examination to identify changes in color, separation, crystallization, etc., in the product's final appearance. And the description should specify the content or label claim of the product. B. Identification. Qualitative identification of the active ingredient in the finished dosage form. And their methods are IR spectroscopy, Raman spectroscopy, and chromatography. C. Assay. The quantity of the drug present in unit weight or volume of ointment. And determined by spectrophotometric method, titrometric method, and microbiological assay. D. Impurities. Impurities arising from the degradation of the drug substance and those arising during manufacturing process of the drug, should also be assessed and controlled. Process impurities, residual solvents, heavy metals and other inorganic or organic impurities. 2. Specific tests. A. Uniformity of weight. Procedure: it applies to products that are not more than 150 grams in net weight. And select 10 filled containers, weigh each container individually and calculate the total weight. Then cut the lateral part of the container and remove its content, then clean the container and dry it to weigh the empty container. After that, calculate the weight content. Weight content equals total weight minus weight of empty container. Then calculate the average weight of content. Average weight of content equals weight of 10 containers/10. Acceptance criteria. 1. Average net content of the 10 containers should not be less than the labeled amount. 2. The net weight of the content of the single container should not be less than 90% of the labeled amount or not less than 95% of the labeled amount. The labeled amount is between 60 grams and 150 grams. If the results have not met the therapeutic criteria, we do the following. 1. Weigh additional 20 containers. 2. Determine the average weight of 30 containers. 3. The net average rate should not be less than the labeled amount, and not more than 1 out of 30 of the content. Note that 90% of the labeled amount should be less than 60 grams and 95% of the labeled amount should be more than 60 grams. B. Particle size determination. This test is necessary to make sure that the particle size is within the monograph specified range. Test technique: take a sample and dilute it with an equal volume of liquid paraffin or glycerol, or, as directed by the monograph. Spoon the liquid onto a glass slide and use a light microscope to observe it. C. Viscosity test. Viscosity measures the resistance of semisolid to flow. At the designated stability test point and at the time of the batch release, the apparent viscosity should be tested. The viscosity test is dependent on the formulation and the process. The viscosity of topical preparation can be measured by using 1. Broke-field viscometer, 2. Cone plate viscometer, 3. Cup and bob viscometer. Let's talk about broke-field viscometer. The preparations are rotated at different speeds and shear rates. At each speed the corresponding dial reading is noted depending on the monograph. D. pH determination. The main goal is to measure the pH to avoid irritation to the skin. Procedure: 1 gram of the sample is mixed with 9 milliliters of distilled water, and the pH of the mixture is measured using glass electrode instrument, as shown in the figure. E. Sterility tests. It is a testing procedure applied to the product and tends to be sterile before marketing to check that this product is free from all living microorganisms. There are 2 recommended methods of sterility testing for pharmaceuticals, and they are as followed. 1. Membrane filtration, which is the most widely used method in industry. It involves pressure or a vacuum filtration of the tested product solution through sterile filtration fitted with a filter membrane. Method of a membrane filtration: first, water is passed through the filter. Second, the filter placed on the surface of a growth medium, example, endo-agar. Third, count the colonies. 2. Direct inoculation, which is small volume of a sample is removed from the sample unit and inoculated directly into suitable volume of growth medium before incubation. Also, it does not require a membrane filter unit. To use direct inoculation, this method not theoretically complex, it requires technical precision in the aseptic manipulation techniques for the proper execution. Also, to use this method it must first demonstrate that the product solution itself does not inhibit growth of typical microorganisms. As a consequence of the repetitive motions involved, it is prone to human errors. E. Test of microbial content. Microorganisms like Pseudomonas aeruginosa and Staphylococcus aureus may contaminate the preparation and, finally, infect the skin. So, ointments should be tested for the absence of such microorganisms. Solutions of different samples of the preparation are made. Each sample is inoculated into separate volumes of half a milliliter of rabbit's plasma, under aseptic conditions, and incubated at 37 degrees Celsius for 1 to 4 hours. No formation of the clot in the incubated mass. 3. Performance tests. A. Spreadability. A sample of 1 gram of semi-solid was placed at the center of the glass plate. Another glass plate was placed over it carefully. Above the glass plate's 2 kilograms weight was placed at the center of the plate. Avoid sliding of the plate. The diameter of the semi-solid in cm was measured after 30 minutes. The experiment was repeated 3 times and the average was calculated. B. Uniformity of container. Carefully remove or cut off the bottom tube seal and make a vertical cut the tube around the upper rim, open the 2 flaps and lay the flaps expose the product. For multiple dose products that contain 5 grams or more, use single tube, remove samples from top, middle and bottom of the tube and analyze. For multiple dose products that contain less than 5 grams of product, if the product fails acceptance criteria A, test 2 additional tubes from the same batch following step 1. And evaluate test results using acceptance criteria B. Acceptance criteria A, all assay results are within the range of 90% to 110% of product label claim. Acceptance criteria B, no assay result is outside the range of 90% to 110% of the product label claim. C. Test of non-irritancy. In this test 24 human volunteers are selected. Definite quantity of ointment is applied under occlusion daily, on the back or volar forearm for 21 dabs. Daily the type of pharmacological action observed is noted. No visible reaction or erythema or intense erythema with edema and vesicular erosion should occur. A good ointment base shows no visible reaction. And, melting point, not less than 11 degrees. Now, let's take some examples of creams and ointments, and their tests at QC. Locoid generic name and formulation, hydrocortisone butyrate 0.1%. Decision rule, Locoid should contain not less than 90% and not more than 110% of the labeled amount. Packaging and storage, it's packaged in tubes or jars. And preserved at room temperature and stored away from sunlight. Identification tests may involve various analytical techniques for Locoid, which contains hydrocortisone butyrate as the active ingredient. Tests like HPLC, IR, and NMR. Labeling, hydrocortisone butyrate, cream, 0.1% is indicated for the relief of the inflammatory and pruritic manifestations of corticosteroid-responsive dermatoses. Brand name is Activate. Generic name, alclometasone dipropionate 0.05%. Decision rule, Activate should contain not less than 90% and not more than 110% of the labeled amount. Packaging and storage, packaged in tubes, jars, or pump dispensers, depending on the formulation. And stored at room temperature away from sunlight. Identification, theses common methods for the identification of alclometasone dipropionate, the active ingredient in Activate. 1. Chromatography. 2. Spectroscopy, IR. 3. Nuclear magnetic resonance, NMR. Labeling, this medication is used to treat a variety of skin conditions, such as eczema, dermatitis, allergies, rash. In the end, thank you for watching this video.